Welcome back to Unit 4. Today we're going to be discussing heredity, chromosomes, genes, and traits. Oh my. So we're going to start with reproduction and variation, then we're going to transition into meiosis, and last we'll talk about chromosomes and alleles. So how do living things have different traits? Variations of genes, which are called alleles, are what give all living things different traits. So variation is dependent on reproduction though. If an organism utilizes asexual reproduction, it only requires one organism, but all of the offspring are identical or uniform. The benefits to this is that it's fast and easy. You only need one person, you don't have to go hunting for a mate and try to woo them over. It's also more efficient. It doesn't need as much energy as far as just the actual ATP and energy cost of reproduction. But the biggest drawback is that there's no variation. For the purposes of our class, we're going to talk about two primary types of asexual reproduction. That's going to be binary fission and budding, and we'll go over those with the videos in your Pocket Lab. Sexual reproduction, on the other hand, requires two organisms, and it occurs by combining gametes, which each have half the number of chromosomes as a regular cell. They're called haploid, is what that is. So this means you get half your genetic information from one parent and half from the other. The benefit of this is variation. You can have a wide range of offspring, even from the same two parents, based on which genes get passed on. The drawback is it does require more energy in general, and you have to have two gametes. There are some species that can self fertilize, but then you do lose that benefit of the variation. So the offspring are diverse because parents provide different genetic material. Remember, each parent has at least two copies of a gene, and they only pass on one. Gametes also undergo two processes during meiosis, which can increase differentiation. We have crossing over, and this is when information can transfer between homologous pairs of chromosomes. So they can essentially trade information across the chromosomes to the same spot. And then we also have independent assortment, which is talking about how alleles are not stuck together. So a gene for green eyes and a gene for red hair don't pass down together. So you can pass the green eyes to one child and the red hair to another. All right, so meiosis is our process of cell division that makes gametes. This is very specifically for gametes only, not somatic cells. And the reason for this is because it has to reduce the genes down to only having one copy of each one. So in humans, we have sperm and egg cells. There can be variation across species, but typically there are two types of gametes within a species, and they are haploid. They only have, in humans, 23 chromosomes instead of the 46. In other organisms, that half number could change, but it's always half. Gametes are formed through what we call reductive division. So cells use PMAT in order to divide. In mitosis, which is how we make somatic or body cells, such as normal cells, it'll only go through PMAT once. But for meiosis, because we need to divide down even smaller with less genetic material in each cell, we're going to go through it twice. So the acronym in PMAT stands for prophase, metaphase, anaphase, and telophase. And we've got our breakdown of that. In the Pocket Lab, you'll have the videos that'll give a really nice breakdown too. But we're going to start here at the 46. We divide, and then we divide again. You can see that division of the chromosomes and the chromatids to get down to that haploid cell. So in PMAT, the same thing happens in each phase. The only thing that's different is that certain things only occur in PMAT 1 versus PMAT 2. So you'll notice here in PMAT 1, with prophase 1, this is where that crossing over occurs. So first stage before chromosomes are lining up and thickening and pairing up with similar chromosomes. And in that first time, the recombination can occur. Then we have metaphase 1. This is where they're all lined up in the middle in a straight line. Now in M1 for meiosis, we have chromosomes lined up in pairs, as opposed to the straight line. because again, we've got to have that double amount, but we'll get there. And then anaphase, they pull away with the spindle fibers, and telophase, 2 new cells are created. In PMAT2, those 2 cells that were just created are now going to split into 4. There is no crossing over in prophase 2 though. They're just going to thicken and start getting ready. Metaphase 2, they're going to line up in a single file line now in the center. Anaphase, they're going to be pulled away by those spindle fibers. And then telophase, we will have the new cells formed. All right, so chromosomes, alleles, and other key vocabulary. Chromosomes are genetic material that carry many genes. They're passed down from parent to offspring. And then you get half and half from each parent. Genes are pieces of DNA that carry instruction for coding, and they're responsible for traits that are expressed. So again, a chromosome carries many genes, and alleles are variations or versions of a gene. So if one gene is for hair color, your allele may be for red, blonde, brown, black. Quick note on that, hair color is actually a multigenic or a polygenic trait, but just to illustrate the idea. All right, genotype versus phenotype. So when we are talking about genetics and heredity, we're going to talk about genotype and phenotype. Genotype is your genetic composition, so gene type, right? Phenotype are your observable or physical traits. So pheno, physical. That's how you can remember this. When we're talking about traits, we're also going to talk about homozygous versus heterozygous. Homozygous is when you have 2 alleles that are the same for a gene. So remember, homo is the root for same, zygous for zygote, which is the fertilized diploid cell. So this means your genotype will have the same allele, whether both dominant or both recessive. Heterozygous is the opposite of that. It's where you've got one dominant and one recessive, or 2 different versions of a gene. We'll get into non-standard inheritance and dominance and recessive states and stuff, but for the most part, heterozygous just means different. So it just means you've got 2 different ones as opposed to 2 of the same. We also talk about dominant versus recessive. So in Mendelian inheritance, we have dominant alleles, and when there are dominant alleles, they will always be expressed when present, and they will prevent the other allele from expressing, though it'll still be carried as a lead, so it'll be turned off. And we show dominance by using a capital letter. So in this case, capital N, that's a homozygous dominant genotype. A recessive allele, on the other hand, will only be expressed if the partner allele is also recessive. So you need 2 recessives in order to see that trait. And we indicate a recessive with the lowercase. I say turned on and off because you always carry the alleles that you inherit from your parents, but your DNA only codes typically using 1 copy at a time. So it's going to use the dominant copy first, and then if something happens to it, or if you've got a bad copy of something, it'll use the good copy instead. I also want to note with this one, the dominant being expressed over all else is only true for Mendelian inheritance, which has what we call complete dominance. Later on, we're going to talk about all other things that can happen with incomplete dominance and other situations where it's not as straightforward.